Hello? I have no idea if this is working or not. Does anyone... I... Wild times. Am I out there in the visible internet realm or not? I have no idea. I mean, I don't know. The thing there says there's an error, but I don't know how to fix said error. The little video is moving around. I don't know if this is working or not, but I shall persevere nonetheless and just pretend that it's working, even if it's not. Welcome to the first Science After Hours live stream. We're not really after hours today. It's 1:30 GMT. Although I suppose depending on if you live in Australia, maybe it's after hours for you there. Not a clue otherwise. But I thought I've been thinking about doing something like this for a while, and I've moved into this new flat here in the sunniest city in Scotland, supposedly, Dundee. And you know, I've got fibre here now. My old flat in Aberdeen didn't have fibre internet. Sorry, I'm just going to text a friend to see if this stream is actually working or not. Just because I have no idea. And I thought if one person can just go and check, then they can, well, they don't have to stay and watch. They can just tell me, yes, no, thumbs up, thumbs down. Anyway, I'm getting off track. Point is that I thought about doing something like this for a while, something to pass the time whilst I wait for my postdoc to start. It is working. Excellent. Look at that. Now you're my #1 top fan. Lucky you. As I was saying, I thought about doing something like this for a while and now have the time and capacity to do it. What to do? What sciencey thing could I possibly be doing on a live stream? You know, most people, they do cool things like play video games and stuff like that, but I don't want to do that. That sounds like a lot of effort to me. I'd have to sort out video games and stuff like that. So I thought instead I would everyone loves a tier list, right? I made a tier list video once. It went on far too long. It was a tier list ranking all the elements of the periodic table. You can go find that on the Science After Hours YouTube channel. But to be honest, I wouldn't recommend it. It's got terrible sound quality. I was just starting out and kept banging the table where the microphone is set up. And yeah, well, that just all came across on the audio. If you're listening to I think it's like an hour or something. Anyway, this super long video about ranking the elements of the periodic table. Also, lesson learned, don't know that much about a lot of the elements of the periodic table. But this tier list video, well, this tier list live stream, I guess it will become a what do the cool kids that do this a lot say? VODs. It'll be a VOD afterwards. It'll become a VOD later, I guess. Is that how it works? Flying by the seat of our pants here, as they say, right? We'll figure it out as we go along. But we're making a tier list video of the organelles of the cell. If you don't know what organelle is, it's a bit like an organ of the human body, but inside a cell, right? So the nucleus, the mitochondria, they're probably the most famous organelles of the bunch, the nucleus being the brain of the cell, the mitochondria being, of course, the powerhouse of the cell. And I found this tier list here. This is all of the organelles that across plants and animal cells, they're all eukaryotic organelles. There's no prokaryotic organelles here. So those would be organelles that are in a bacteria. But we're not talking about bacteria today, talking about animal eukaryotic cells, plant cells, which are also eukaryotic cells. So just your stereotypical eukaryotic cell that eukaryotic are made out of, and your plants if you are growing any. We have a bunch of tiers here from S to F. Obviously, everyone I think in 2025 knows how a tier list works. I have no idea how long this will go on for. This stream could be 15 minutes if I have very in-depth, opinionated opinions on organelles and how good I think they are and where I think they belong. I've had to zoom in my screen a fair bit when I was testing this all out. These little isn't an opinion if you are right. That's or is it just a fact? What is the difference between an opinion and a fact when it comes to science? But the point is, these little tiles, widgets, the things with the names of the organelles on were really, really small. So I've had to zoom my screen right in. So now you can't really see all of the tiers at once. But when we get to the end, we'll just zoom out and we can view our handiwork. Celine, you're a data lady, so we shall see if we get a normal distribution amongst our organelle selections, or if I inherently have some sort of bias towards what sort of organelles are good and bad. But we'll see. Well, let's just look at organelles. So the first one, at least according to this, is the nuclear membrane, as you might imagine. Well, that's true, but there's only 1, 2, 3, 4, 5, 6, 7, 8, 16, 23 options here. So once we get to 23 options done, if I put them all in the F tier, for instance, then there's no normal distribution winning out of that. But again, what do I know? I'm just a wet lab scientist. That's on my basic understanding of the normal distribution and bias and whatnot. If it's not a bell shape, then it's not normally distributed, I think. I hope. Squishy science, wet science, they're all one and the same. Look, you're here. This is Science After Hours. Should I rename the channel Squishy Science After Hours? But I fear that may sort of draw in the wrong crowd. People wanting squishy science videos. I've got my undergraduate lab coat on. I thought I'd get some sort of use out of this. We had to buy our lab coats in our undergrad. This isn't the one from my PhD, so it doesn't have salmonella on it, so you don't have to worry about that. It does have some stains on it. There's I think what looks like a hematoxylin and eosin stain on the sleeve here, which is a stain for cells. It stains different membranes. But well, you wouldn't have lab time in your undergrad. Maths labs? You're doing MATLAB coding sessions, graph drawing. Is that a lab session? You ought to sit round and draw a nice graph, a sigmoid curve. That's a type of graph, right? Or an exponential. Anyway, this is my undergraduate lab coat. I thought, well, if I'm doing a live stream, why not dress up for the occasion in a sciencey way? This is the best I could come up with. I had time in the maths corridor. These are the darkest hidden corridor on the top floor. What were you doing in the corridor, though? Just hanging out with all the other mathsers, the mathematicians. You're just talking about graphs and mathsy things, right? So being lonely and miserable, is that it? Well, science is a team game. Maths is not, I think. I don't know. How do you do an equation as a group? Sit round. Oh, I think the x should go on this side of the equals, and let's do an integration or something. I don't know. Is that how team maths is done? Whereas science, it's collaborative effort. One person does this part of the experiment, another person does a different part of the experiment. You pool your data together, and you've got the whole picture. But maybe the longer I speak, the less likely I will be to work at a university and make friends with mathematicians. So don't worry, Celine. I've got one mathematician friend, and you can put up with all my derogatory comments towards you. Well, not towards you, I suppose. Towards maths. I use maths for dilutions. It's not as if and stuff like that. But usually, I have one equation. C1 V1 equals C2 V2, and you can repurpose that for almost any endeavor you could possibly want. How much salt should I put in this more salty water I'm trying to make? How much agar should go in this agar plate mix? C1 V1 equation. Excellent equation. But let's talk about organelles. Or at least, let's talk about at least one organelle before we go off on some other tangent. I'll try and not make it about disdain for mathematics. So the first organelle here is the nuclear membrane. That's, of course, the cell membrane that goes around the nucleus. Does it really have a function beyond separating the nucleus from the rest of the cell? Yeah, I suppose. I mean, it's got pores in it that allow material to travel backwards and forwards, but it's not the most exciting of organelles. It's not out there causing exciting cell functions or whatever. And in fact, if you think of, I know this is eukaryotic cells, but it clearly isn't that crucial, because I'm sure someone now that researches nuclear membranes is going to be like, you're wrong. But in a prokaryotic cell, a bacteria, they don't have a nuclear membrane. Their DNA just free floats in the cytoplasm, and they can get on just fine. So I know we have a much larger genome than your little average bacteria, but I think that suggests to me that the nuclear membrane is probably not the most exciting of organelles. I'd feel a bit bad putting the first organelle all the way down here in F. So D. Mine, Daniel Underwood, that starts with D. D for the first organelle. It's all meant to be. But the next one here, down here at the bottom, is the cytoskeleton, and I'd say that's much more exciting. You wouldn't have a cell without the cytoskeleton. It's think of you as a human being. If you didn't have a skeleton, you'd just be nothing, just a bag, a nebulous bag, a pool of liquids and skin and organs on the floor, not being able to hold together. So, I mean, you haven't done for a while, to be fair. You've kept your body in fairly solid condition for a while now, which is for you quite an achievement. So congratulations. Cytoskeleton. Without that, the cell, I mean, a cell is really just a bag, but the cytoskeleton at least holds this bag in some sort of structure together, and I think that's very important. The cytoskeleton is also important for moving things around inside the cell. So, dragging things and pulling things and changing the cell shape for all sorts of needs. So I'm saying the cytoskeleton is an A-class organelle. Spoilers for later. I mean, I haven't thoroughly studied all the organelles in this tier list. I sort of just found a good tier list and thought, hey, let's stream that and see what happens. What on earth is that noise outside? I don't know if you can hear it, but it sounds like, I don't know, a giant bee is attacking the local area of Aberdeen. I don't live in Aberdeen anymore. This is Dundee, or Fundy, as you say there, Celine. Although in Aberdeen, people call this place Scumdee. And again, I guess really making new friends here. Dundee's not that bad. At least the bit of Dundee that I have been to thus far, it's just fine. It's a city. I lived in London, for a period of time when I did my master's degree, and I hated that. I mean, I've only lived here for a week, and I can't say I hate it yet. Although, to be fair, did I hate London when I only lived there for a week? No, probably not. The excitement of London living was, probably still in full swing. I also lived in King's Cross, which was, slap bang in the middle. It's full of people everywhere and litter and people and tourists and whatnot. In this part of Dundee, whatever this bit's called, Stobsy, Stobswell, Stobs something. I can't see any tourists here. Paris also has a litter problem, don't you think? I mean, it's been a long time since I've been to Paris, but from what I remember, big litter problem. So, Dundee, Aberdeen, yes, there is litter here, like all cities, but it's not. You can go 5 minutes without stepping on a crisp packet on the floor or some other thing, So that's a thumbs up from me. Less litter equals better city. That's what I say. Well, maybe you should move. I know you live for the cheap rent, but pay more rent. Come live with me. I've got a spare room. Look, you can be right here. There's the spare bed is literally down here next to me. You can just lie here and not be surrounded by litter. It's fine. Move to Italy. Andre won't mind. Live that not Costa del Sol, because that's in Spain, but Costa del Italy. Hey, I was inviting you into my spare room, in a nicety. Don't make me out to be some sort of felon. I am a stand-up member of society. I've got a lab coat on, and scientists are known for being quality people. There was that guy that did that sort of illegal genetic modification on those kids so they wouldn't get HIV. That was quite a few years ago now, but stand-up member of society. Organelles. Let's just go back to organelles. The nucleolus. The nucleolus is the center of the nucleus. It's that sort of dense core inside of the nucleus. That's sort of where all your genetic material is, and sort of that really is the heart of the heart, not the heart of the cell, the center of the brain, right? And that's important for cellular functions. But other than that, I mean, I'm not a nucleolus expert. Much like, I guess, my opinion of the nuclear membrane, I don't think it's that important. I think it's more important than the nuclear membrane because being inside the nucleus, that's where your genetic material is, rather than just surrounding it and allowing things to pass backwards and forwards. So I'd say the nucleolus can go in the C tier. For all you nucleolus experts out there, please feel free to leave a comment and complain. And give me some papers to read about the nucleolus. I'm an immunologist, really. And look at, so when it comes to nucleolus, not really relevant for my host pathogen interaction research, or at least it hasn't been as of yet. Who knows? About to start a new postdoc job. So maybe I'll find out in a week or so that the nucleolus is crucial to my future career development. But at this moment in time, I've never read a single paper about the nucleolus, I don't think. But a thing I have read many a paper about is the cell membrane. It is you've got this double the phospholipid bilayer, right? I'm sure you all learnt that when you did biology at school. At least I certainly hope you did. Is it is allows for the diffusion of things to pass through it. It's osmotic. It can be selective. It's a semi-permeable membrane. Well, that's because we're old. But you can think that we're here now. You're relearning. It's like you'll feel young again by the end of this. You'll be like, wow, I've relived my youth, and now I want to be a biologist. I'm going to give up doing this maths, boring maths, coding, and whatever else you do, data analysis. So the cell membrane is, that's basically, without cell membrane, you wouldn't have a cell. Cell membrane breaks, cell dies. The cell membrane is important for, say, the passage of materials backwards and forwards from the host to the cell and from the cell back into the host. It's important for microorganisms. They can latch on to the cell membrane and invade the cell. It's important for viruses, for instance. They can bind to the cell membrane, enter the cell, and then when they want to leave that cell and continue their spread throughout your body, they make use of the cell membrane. So I say the cell membrane, that is an S-tier organelle. I mean, is it more important than a cytoskeleton? Yes, because if you don't have a cytoskeleton, you've got sort of like a wet bag with no rigidity, that you wouldn't have a functional cell. Sure, but you'd have something. Without the cell membrane, that liquid, the cytoplasm, and all the stuff that's inside the cell that makes up the cell would just disperse into nothingness. You need that cell membrane, and so yeah, I'd say that's an S-tier organelle. You can't do any better than that. Right, what else we got? A centriole. That's a rogue choice, and I'm not entirely sure if that's an organelle, to be fair. I'm going to go to my other tab and have a little cheeky Google, because I think the centrosome is the organelle, whereas the centrioles are just sort of microtubule tubes. Centrosome and centrosome. Let's have a look. Yeah, in cell biology, the centrosome is an organelle that serves as the main microtubule organizing center, the mTOC of the animal cell. So that means to me that centriole has to go in the F tier. It's not even an organelle. It's a component of an organelle. Sorry to be the bearer of bad news to all you centriole fans out there. Why is someone mowing their lawn? I'm trying to be a famous YouTube science streamer now. Should you be? It's 2 p.m. almost. Should you be at work or something? I know you could argue that I should be at work as well, but my contract hasn't come through yet. Also, it's not that sunny, so that doesn't seem like good weather to be mowing the lawn, if you ask me. Then again, I'm not a gardener either. That was my parents' thing. I've lived in flats for a decade now. I haven't had a garden for a very long time. So, whilst that person is mowing the lawn, let's talk about the cytoplasm. The cytoplasm is the liquid that makes up the cell. It's the liquid, the main component of the cell. It's probably the largest organelle, because in terms of volume at the very least, because whilst your plasma membrane, your cell membrane, is covering the cell, that entire liquid component that is inside the cell is all cytoplasm, and that cytoplasm contains all your other organelles, your mitochondria, your nucleus, your centrosome, your nuclear membrane. That's all in the cytoplasm, and all the reactions within the cell take place in the cytoplasm in some fashion, or use the cytoplasm in some way. Even things that are made in the nucleus and around that nuclear membrane, like mRNA, that's the thing that will then lead to your genes being translated and proteins being produced and life existing. That translation is taking place in the cytoplasm. So the cytoplasm is where all your enzymes are, all your RAB GTPases, and my protein of choice, P62, sequestrazone 1. That's what I did my entire PhD on. That's in the cytoplasm as well. But is it as important, in my opinion, as the cell membrane? That is the tone of that lawnmower, really bothersome. It's just that slight pitch that I find quite annoying. So that's a tad off-putting as we decide where to put the cytoplasm on this tier list. Do I put it in the S tier? Is it as equal to cell membrane, or is it more like the cytoskeleton? I don't think it's a B tier organelle. It's definitely an S tier or an A tier organelle. Do I put them, they both have the word cyto in them. Do I just put them together because of that? But then the cytoskeleton is in the cytoplasm. You know what? S tier. I'm going to put an S, the cytoplasm, along with the cell membrane. It's a crucial component of the cell, although one could argue that they're all crucial components of the cell, and that's why it's evolved that way. But the cytoplasm, more crucial than most. So S tier organelle. Now, the lawnmower has stopped. Do I get up and shut the window? Because it gets quite hot in here. Or do I not? Maybe the lawnmowing has stopped now. Problem is the angle of the window is I can't see out as to whether the person is still going to be mowing their lawn. Maybe they've just finished. Are they emptying their grass? Or are they like, oh, I just need to drink a cup of tea and then I'll get back to it. I'm taking the risk. I will not close the window. The sounds of Dundee can be heard in the background of this first live stream. And, I'll be pretty annoyed if the lawnmower starts back up immediately, but I certainly hope he can't hear me, or she, or they, of course. I don't know who is mowing their lawn. How very sexist of me to presume it's a guy. Whoever it is mowing their lawn, I hope they can't hear me. But if they can, maybe they'll learn something about organelles and my opinion on certain organelles at the same time. So if we go back down to the bottom here and look at our selections, the endoplasmic reticulum, the rough endoplasmic reticulum in particular, is next up for selection. Well, now I figured out what that noise is. It's a strimmer. There's some hardcore gardening going on outside in the Stobbsy area of Dundee today. So whilst they're doing their gardening, they can learn about organelles, and you can learn about organelles too, whether you're Stobbswell. I wasn't that far off. I knew it was Stobbs something, so I'd take that as a, I've only lived here a week. I'll take that as a victory for me. So well done me. The rough endoplasmic reticulum. This is the endoplasmic reticulum, bits of cell membrane involved in cell trafficking. But the reason it's rough is it's got ribosomes on it. Ribosomes, of course, they're important for translation, turning that mRNA into functional protein from your, getting those tRNAs up in there that got amino acids attached, all that translation that I hope you learned about when you were doing your AS biology, Celine, or hopefully before. I'd hope you knew what transcription and translation were before A level, but I don't know what sort of schooling you might have had, whether it was good or not. But the rough endoplasmic reticulum, you wouldn't have proteins without it because it's got the ribosomes on it. But it was good for what it was. I mean, that that could be read in many ways. If it was like a school for delinquent children or whatever, you could say it's a terrible school, but it was good for what it was. Oh, it was a Catholic school, but we did do the squishy science, right? How many how many classes did you have on the old evolution? Did you cover much of that, or was it one of those really devout Catholic schools where you can't talk about such things? We all came from Adam's rib bone, or however the story goes. Now not only have I upset mathematicians, I'm now veering onto upsetting religious folk as well. I have a GCSE in religious education, to be fair. It was a compulsory thing at my secondary school, so we had to do it. It was, to be fair, quite an interesting GCSE. It was like a comparative thing. We didn't just have to focus on one of all religions or whatever. It was specifically focused on Hinduism and Christianity and the comparing and contrasting between the two. I did all right. I got an A in it. It was also a good GCSE for improving one's writing skills because the exams were all essay based. So if you couldn't write an essay well, you wouldn't do very well in that GCSE. We also went on a school trip. Went to a mandir once. They had good snacks, if I remember correctly, at this mandir. So I remember that. So I guess maybe we also had a class once. Our religious education teacher, Mr. Brown, he went on. We were talking about going on, what do you call it, pilgrimages, right? We were talking about pilgrimages, and he personally went on a pilgrimage to the Joshua Tree from the U2 album. He went to the actual Joshua Tree and almost got lost in the desert and died or something, from what I remember. I mean, that was almost 15 years ago, that class. I think that's what happened. He went and searched for this tree and then got lost in the desert. We were taught by a nun. I mean, I've seen Sister Act, so was it like that? Was there a lot of was it like Sister Act before or after Whoopi Goldberg joins the convent? Right, so she was scary. So before Whoopi Goldberg joins the convent. Well, religion is not my bag. If it works for you, that's totally fine. We all have our strong beliefs, and that's fine. You can do whatever you want, but not for me, and that's totally okay. But the rough endoplasmic reticulum, say it's got those ribosomes on it, so that's necessary for protein production. It wouldn't again exist without any proteins. But I mean, I can't really use that as a justification because you wouldn't really exist without any of these organelles in some fashion. I mean, I say that, but they're nuclear membrane. They're not really present in bacteria. But then the rough endoplasmic reticulum is also not present in bacteria. The ribosomes in prokaryotic cells are free flowing. Now, I know the endoplasmic reticulum in the eukaryote has got a lot of other uses. There's a lot of transport and stuff that goes on there, but I can't all put them up in the S tier. I mean, do I think the endoplasmic reticulum, the rough endoplasmic reticulum, is as important or as cool? I mean, what is making an S tier organelle? Just how much I like it, how much I know about it, how crucial it is to the cell. I feel like these two are up here because of how crucial they were to the cell. But again, using that logic, they're all going up in the S tier. I'm going to have to. Do you know what? I'm feeling just out of gut. The rough endoplasmic reticulum should go in the B tier. I don't know what it is. It's just what the science gods are telling me today, that the rough endoplasmic reticulum is a B tier organelle. It's getting those points for the ribosomes being on it, but other than that, yeah, it's just a bit of trafficking and a bit of storage and not much else. But the nucleus, well, might as well just put that straight in the S tier, right? That is the brain of the cell. That's where your DNA is. It's where your chromosomes are. Obviously, what DNA is stored in. If you don't have a nucleus, your cell is not going to be able to do much. I say it won't do nothing, because some cells don't have nuclei as a space-saving measure. So that's important. But obviously, if all your cells didn't have nuclei, you wouldn't exist. Nothing would be happening. You'd have no DNA being produced. And therefore you'd have no transcription, and then no translation, and therefore no proteins, and therefore you'd have no enzymes, and then nothing would happen. Life would not exist without the nucleus, or at least DNA in general. And so as I am conflating the nucleus with DNA, because again, prokaryotic cells, their DNA is just free, so it's not really a nucleus per se. But I'm saying the nucleus is an S tier organelle, no doubt about it. It's going up there at the top. Endosomes, well, they're little. Oh, good. What sort of tea? What sort of tea do you have? I know it'll be without normal milk. I'll tell you that. Was it Earl Grey breakfast tea? Green tea, fruit tea. Elderflower. That sounds absolutely vile. No offense. What's wrong? When you said you were off to get tea, I was imagining, builder's tea, normal tea. Elderflower tea. I mean, I don't mind, I don't know, drinks with elderflower in, right? And some sort of elderflower cordial or something. Apple and elderflower, that's a good combination. But elderflower tea, you don't have any oat milk. Well, get some. I don't know how you make oat milk, because I mean, just get some oats and buy. Oats are fairly cheap, right? So you can make your own oat milk, perhaps. That's another thing, I've devoted my life to learning how salmonella interacts with the macrophage when it comes to other things going on in the world, like how is oat milk made. Couldn't even give you the slightest idea. I presume it involves getting oats wet in some fashion, but how that then leads to milk in quotation marks. Okay, so it's elderflower and strawberry. So it's not just elderflower. I mean, I'm not a fruit tea guy. I think the fruitiest of teas that I enjoy is an Earl Grey, but I tend to stick with, your bog standard breakfast tea, or your builder's brew, as you so wonderfully put it. But learn how to make oat milk and then report back to me. And then if it's easy, I can make my own oat milk. Maybe we can go into business. We can call ourselves it's blended oats and water. Oh, is that it? Oh. Well, that's that's karma for me there. Oh no, it's back. Was that it? Blended oats and what? You just mix the two together in and that's it. Oat milk. I presume you must run it through some sort of sieve, otherwise you've got bits in your oat milk, but I mean, I'll give that a squeezing through a cheesecloth. Okay, this is getting more effortful. I was hoping it'd be something, like the things that you get when you make cocktails. The thing they pour a cocktail through at the end that's a bit like a really fine colander. I thought that might be good enough. Maybe I could make oat milk in the cocktail shaker that I have. I've got a lot of free time whilst I wait for my contract to come through for this postdoc job. So maybe I'll give that a go. Endosomes. Endosomes are little vesicles that travel out of the cell. No, that's an exosome. A endosome travels within the cell. It's a bit like a vacuole, I suppose, a tiny little vesicle that's got stuff. It's got stuff in it for trafficking stuff around, moving things, sending messages and whatnot. That's quite important. It's allowing bits of the cell from within the cell to communicate with one another. So I'd say that's fairly important. But overall, endosomes just little. They're like envelopes, and envelopes are not that exciting. So an endosome can go in the middle. It's not a bad organelle, but is it an amazing organelle? No, it's not. It's a bog standard, right down the middle organelle. C. Chloroplast. Oof, plants. That's a plant-specific organelle. That chloroplasts. They're the green things in plant leaves that allow for photosynthesis. Now this is going to show my disdain for plant science. Not that plant science is not important, it's just not for me. I'm a cell biologist and immunologist, sure, but on mammalian cells, really, and bacteria. I've never once studied a plant cell. I have no intention to study a plant cell. The last time I had to think about chloroplasts was probably back when I did my A-level, learning about the Calvin cycle and how sunlight from the sun is used to make energy, and that's what the chloroplasts are for. I'm not going to put it in the F tier because the centriole is in the F tier, and that wasn't even an organelle. So the chloroplast is better than something that's not even an organelle on this organelle tier list. But I'm not a plant scientist. For people that love plant science, good for you. You have my full support, because I say it's more of an acquired taste than your typical cell biologist science. So good for you. You're doing important work out there, but it's not for me. And so a chloroplast going in the E tier. A vacuole. Oh, we've got different types of vacuole. Well, that's fun. Okay, well, let's start with basic vacuole. It's got the word basic in it, right? A vacuole, it's like a bigger sac than an endosome. Basic vacuole, again, it's just got stuff in it. Is it the most exciting? No. Is it that important? Well, yes, but again, it's in the cell. They're all important. I feel like a vacuole can go in the C tier. You can be as childish as you want. This is a free space. We may be talking about some exciting science here. Ranking the organelles of the cell. Just what people want to be doing on their what day is it today? Thursday afternoons. Be fun. Be childish. The Cineworld, when you go to Cineworld, this is a real tangent, right? If you're watching a family movie, something like, I think the last family movie I went to see was Pixar's Elio. Probably one of the few people that did go and see Pixar's Elio. But at the beginning, when they're going through all the adverts and stuff, they have a special sting for family movies, and it says "Be more childish" on it. Encouraging. I think it's for the family ticket thing. I think one adult goes free with 2 kids or something like that. I don't know. Look up the Cineworld website. I am not a spokesperson for them. But anyway, they've got some deal for families, and trying to encourage adults to go and watch these kids' films, they have a little sting. It says "Be more childish." Well, think of me as your Cineworld for science this Thursday afternoon, and be more childish. It's absolutely okay. No complaints from me. Don't be offensive or whatever. This is my first stream. I don't want to be banning anyone. Don't make me get a moderator or whatever people do on streams to stop such vile behaviour. Well, good you are, but yes, you're behaving at the moment. But some children, remember that show? Uh, what was that show? Super Nanny. Remember Super Nanny? Do they still make that? I don't think so. Of Super Nanny, it was on Channel 4, right? Those were children being childish, but they were horrible kids. I don't want to have to go all Joe Frost up in here and make you sit on the naughty step for viewers. Because I haven't got time for that. I'm busy thinking about how good I think certain organelles are. Now, a food vacuole, well, that's obviously important for nutrients for the cell. So I would argue then it's probably a bit more exciting than your bog standard basic organelle. So then do I put... No, I think the food vacuole should go in the B tier, right? Because I don't think an endosome and a vacuole, they are, a bit dull, right? So C tier is the a bit dull tier, right? If you get a C in a test, you've done just fine, average, right? You haven't failed, you've just passed, right? So you've done the average. If you watch a film and the cinema score for it is a C, you're like, yeah, it's probably just fine. So the C tier organelles, just fine. But the food vacuole then should be more important than the basic vacuole, and therefore I think should go in the B tier along with the rough endoplasmic reticulum. Then we've got this contractile vacuole as well, which I guess should then go alongside the food vacuole. Do I think vacuoles in themselves aren't that exciting as organelles? So I don't think they belong up in the S and A tiers. They're just big sacs. They're sacs within a sac, I suppose. You can imagine your cell it's like, if you imagine your cell as a Christmas stocking, right? And then inside of your Christmas stocking, you've got wrapped presents. Well, the wrapping of those gifts inside the stocking, you could argue, is vacuoles. And these vacuoles, much like a Christmas stocking, they can have food in for nutrition for you, probably a Terry's chocolate orange or some other bog standard Christmas snack, a Twiglet tub, or they can have function, i.e. toys or socks or deodorant or perfumes or something like that. That'd be a contractile vacuole with a function. Not that delivering food is a function. I know, but yeah, it is what it is. Next up, smooth endoplasmic reticulum. Well, that's the same as the endoplasmic reticulum, but without ribosomes on it. So I think the ribosomes give the rough endoplasmic reticulum a little bit of a leg up. They've got a little. It's endoplasmic reticulum, but with a fun thing. There's something in this lab coat. I just put it on before this stream, so that was a surprise to feel. What on earth is this? Oh, well, I'm going to put this on. The smooth endoplasmic reticulum, whilst I do this, can go in the C tier. It hasn't got that extra wow factor of the rough endoplasmic reticulum. Yeah, it's a name badge. I don't know where this is from. This must be, I finished my undergrad in 2018. So this thing here, it's 2025 now. It's been in this pocket for God knows how long. Probably from an open day or something, I imagine. Exactly. No, it wasn't in case I forgot my name. It was so other people could know my name. And then, so when they, when I go on to win a Nobel or whatever for exciting science work that I may do, everyone, I can whip out this badge and wear that to the Nobel ceremony. so that everyone knows that I was the Nobel laureate for science that year. Let me. It's quite a rusty pin. It's clearly been in there a while. This is probably exciting viewing for the people watching at home. There we go. Look, can you even see that in the stream? Probably not. But you know, maybe I can sort of up my pocket up here somehow. My eyes aren't that good. Well, my eyes aren't that good either. Maybe the badge was for me, so I can be like, oh yes, that's me. You know, this is like having a badge is when you're. We didn't have lanyards in my undergrad. You know, we had the little identification card things, but they weren't on lanyards. So one could argue that having a badge is sort of like the first stage before you get a lanyard. And now, as a real scientist, you get a lanyard, and then going from there, looking around at my desk here, you get fancy lanyards. This lanyard here is from Merck. It's got my old University of Aberdeen badge on it, but it's also got this like stretchy thing, so you don't have to, you know, drag your neck out to touch things. It's got this like fun little elastic thing. It also makes it a better weapon, and if I was to become some sort of, I don't know, assassin, I could use this as a garroting wire to kill my scientific enemies in the lab if I'm there doing an experiment and I think, oh, this colleague of mine is going to touch my pipettes and crank them too far and break them. I can whip out the cord and sort them right out. Or maybe if I think they're going to scoop me and publish some data that's similar to mine before I'm ready to publish, I can put a stop to that and knock them out with the little cable. No, this isn't happening. Well, look, I was going to say I'm glad you left Squishy Science too, but then I realised that comes across as quite insulting. I meant it in the case that, you know, not everyone, and that includes me, likes analysing data, big data sets. You know, there are specific people trained to do that. You know, we have like bioinformaticians and general data analysts. Someone has to do that horrible job. And you, Celine, you have taken up the slack to be able to, you know, have those skills. Do the things that other people don't want to do. And for that, you should be very proud. The Golgi apparatus that's that fun-shaped organelle. If you ever looked at a picture of the inside of a cell, you know, it's the one that's made out of different layers. Again, it's another organelle that's important for trafficking. Vesicles go there to get sorted out, packaged in new ways, and then sent off to other parts of the cell. I think it should get bonus points for its fun shape. There's no other organelle with the structure like the Golgi apparatus. Its function, not the most exciting, as I say. It's just for packaging and trafficking and whatnot, but it's got such a cool, funky shape. I feel like it deserves to go in the A tier. And as I'm looking at this, how this thing is unfolding, I feel like, wow, maths is real. This is developing somewhat of a normal distribution without me even really trying. But now I've noticed that it's a normal distribution. Do I just, you know, purposely skew it just to be me about it? You know, do I want maths to win? No, that is not very ethical, is it? I should uphold my scientific ethics, and the data is what the data is, whether it's normally distributed or not. I can do some sort of non-normal distribution test, Spearman's rank, something like that. Spearman's rank, not normal distribution. I can't remember. Not a t-test. Can't do a t-test or an ANOVA. Got to do something like a spearman's rank or a chi-squared. Is a chi-squared a non-parametric test? You can do a t-test on normally distributed data. Yes, that I know. I'm trying to think non-normal distribution statistical test. Spearman's rank, right? Spearman's rank is the t-test equivalent, or that's a Mann-Whitney U. A Mann-Whitney U is the equivalent of a t-test. God, now this is something I really should know. I think it's a Mann-Whitney U is the equivalent of a t-test. I think I used it in my PhD on some of the data that wasn't normally distributed. Well, it's too late now. They've already given it to me. Spearman's rank is more for correlation. Oh yes, Spearman's rank is correlation coefficient. You're absolutely God. Oh, I need to go back in the lab and do some real science. My mind is failing me. Hope my new boss doesn't watch this and they're like, oh, he doesn't know anything about statistics. Why on earth are we going to hire this guy? Yes, imagine that. Well done. Well done. Your mathematical training has set you up for my downfall here this afternoon. But I am right about Mann-Whitney U, aren't I? A Mann-Whitney U is the non-parametric t-test for comparing two averages. Right? Well, whilst you're deciding whether whilst fate comes my way, let's look at the peroxisome. Peroxisome is non-parametric. Yeah. Well, thank goodness for that. Maybe they will let me keep having my my PhD. I like an ANOVA. I know you don't often get to use an ANOVA in the stuff I do because you only really have two groups, but like an ANOVA. Cool name. You get to do post hoc tests, the Tukey test, which is also a cool name. You have to write ANOVA in all capital letters. No, it's more fun than t-test. But t-test is, the workhorse of statistics. I'd say at least in my field. But the peroxisome, it's an endosome, as you might have guessed from the fact it ends in an ome. It's important for immune response. It's got peroxides and stuff in it, as you might have guessed from peroxisome. It's important for the destruction of stuff, helping destroy things. When you start writing about a two-tailed, two-hypothesis test, that is when it gets fun. See, I never understood this tail thing. I know it shows directionality of where you imagine your data to be, right? I know if you do it in two-tailed, you're saying that your difference is above or below, or if it's only one-tailed, you're adding a bias because you know the directionality of your data. Is that right? I think that's right. It's about the tails on the bell. Yeah, I'm making a rod for my own back here by talking about things I think I understand, but could be wrong. But then I guess this live stream will be here on the internet forever, and everyone else can learn about statistics as well as organelles. Yeah, the tail on the bell curve, it determines your success rate. Well, I have a success rate of 100%. I've decided. I mean, this stream is working, and that's a 1 out of 1 streams. So 100% success rate for me so far. Peroxisomes, they're more fun endosomes because they destroy things. So, they need to go above the endosomes down here. Are they more exciting than vacuoles? Again, I think the destruction aspect makes them more exciting than a vacuole. So they can go up here in the A tier. I don't think they are as fun and exciting as these three in the S tier, so they're not getting up there. Peroxisomes, along with the Golgi and the cytoskeleton, you can stay in the A tier. We've only got 1, 2, 3, 4, 5, 6, 7 organelles left, which means the stream will be over soonish, maybe, unless we get sidetracked by more fun statistics chat or whatever else might come along. But lysosomes, well, I'm going to show my own bias here. Lysosomes are going in the S tier. Lysosomes are the organelles that are ultimately responsible for the destruction of invading pathogens. As someone that works on host-pathogen interactions, the lysosome is very important. Bacteria have special abilities to disrupt the lysosome in some way, whether that prevents the lysosome from destroying the bacteria or somehow tricks the lysosome into making the environment for the bacteria actually more hospitable for the environment. Lysosomes have a very low pH, and some bacteria rely on that reduction in pH from the neutral cytoplasm, like pH 6.57, to the acidic pH of the lysosome, that's pH 2-ish, 1, 2, to kickstart their survival and reproduction and whatnot, and the activation of all sorts of bacterial cell pathways. And I think that's really cool, and that's why the lysosome goes in the S tier. Less about the function of the lysosome in the host cell, I suppose, but more about how the lysosome is utilized by pathogens for their own benefit. That's that sort of thing where it's how different organisms have evolved to be able to survive against one another for their own benefits. That's one of the most, I love that sort of thing. That's why I like science so much. The nuclear plasm, well, I mean, that's like the cytoplasm, but in the nucleus. I don't really have that much to say about it. I feel like it can go in the D tier along with the nuclear membrane. It's not, I don't have disdain for it like the chloroplast, and it is an organelle, I suppose. So it doesn't really need to go in the F tier. Although, would you consider the nuclear plasm an organelle? Let's have a look. Nucleoplasm. Let's see how this goes. Nucleoplasm is quite similar to the cytoplasm, with the main difference being that nucleoplasm is found inside the cytoplasm, and while the cytoplasm is inside the cell. But is the nucleoplasm an organelle? Nucleoplasm, organelle. Let's go on this journey together. The nucleoplasm is a type of protoplasm. Is the nucleoplasm an organelle? The human cell in the nucleoplasm. Not really seeing the word organelle anywhere, so maybe it should go with centrosome in the thing, centriole, whatever it was. Not seeing the word organelle. Let's go on Wikipedia. I know you shouldn't really use Wikipedia for such things, and just look for the word organelle. That makes up the cell nucleus, the most prominent organelle in the eukaryotic cell. So that's saying that the nucleus is the organelle and not the nucleoplasm. So, you know what? Sorry, nucleoplasm, you're going down here with centriole, because you're part of an organelle. You're not an organelle in your own right. So you're going down here with centriole. That was part of the centrosome. Ribosomes. Well, I suppose that's up for debate as to whether they're an organelle or not. I mean, I suppose you could say yes. They're necessary for that protein construction that we talked about earlier. It's on the rough endoplasmic reticulum. I feel like it would be a bit disingenuous to separate out the rough endoplasmic reticulum and ribosomes, so they can both go in the B tier. Put those next to each other, why not? They're so important for each other's functions. Cell wall, well, that's a plant thing in this instance, because this is from eukaryotic organelles. Now, I know I talked about how some of these things got bonus points or minus points for how they are in prokaryotic cells, but your mammalian cell does not have a cell wall. It has just your cell membrane, and that's it. Whereas your plant cells, they have this cell wall around the cell membrane to provide extra rigidity, and the same is said for prokaryotic cells. But I don't like plant cells. I think I've made that clear. That's not my thing. As I say before, just to make sure, if plant cells are your thing, great. No shade from me. We all research what we love. I'm sure salmonella and host pathogen interactions is not someone else's cup of tea, and I would expect such disdain for the lysosome or something down here. The mitochondria, well, there's no point arguing about that. It's the powerhouse of the cell. It's the organelle that people know the most. Everyone's like, oh, what's the mitochondria? It's the powerhouse of the cell. It makes your ATP. It's important for sorting out reactive oxygen species. It deals with electrons. It's a very complicated organelle. It originated, the theory goes, that was originally a bacteria that was internalized into a cell, and then they form some sort of symbiotic relationship together. They relied on each other. The cell that then eventually became a eukaryotic cell then got bonus respiration from these bacteria. So they then, they started to live together, but then it was no longer necessary for this bacteria to be a whole bacteria, and so it has changed over time and is now just the powerhouse of the cell. Most well-known, beloved organelle is the mitochondria. Vesicles, well, vesicles can go here in the middle with endosomes and vacuoles and other stuff. They're just little bags within bags for trafficking stuff around. Nothing too exciting there. And then last but not least, chromosomes and chromatin. Obviously, so that's where your DNA is. That's how your DNA is stored. You wouldn't be a functional organism without DNA. So for that reason alone, it should probably go in the S tier. I mean, you wouldn't have any of this other stuff if it wasn't for your actual DNA. So a chromosome should go in the S tier, I suppose. And let's take a little zoom out and see what we have done here. I mean, it's not quite a normal distribution, is it? It is skewed in this more positive direction. Hello, welcome to the stream. We're looking at organelles today. I mean, we've just finished the ranking of all these organelles, but so we're saying that this organelle distribution is more positively distributed. We've got more things up here than in the center. It'd be worrying if it were perfectly distributed. Would you say so? I feel like, tier lists are the perfect example to find normally distributed data, wouldn't you say? I mean, although they're based on opinion, I suppose. An opinion is natural bias. So that's going to introduce some sort of skew to the data, right? And therefore you're going to end up with distributions like this. Let's have a little review. We'll start at the bottom. So these F tier organelles were not organelles. That's why they ended up in the F tier. The nucleoplasm, part of the nucleus, not an organelle, and the centriole, part of the centrosome, not an organelle, F tier. E tier organelles are the chloroplast and the cell wall. Because I'm not a plant scientist, they get a lot of minus points just for that. If you're a plant scientist, again, sorry, didn't mean to upset you. Nuclear membrane was the only organelle in the D tier, all sad and alone by itself. But I just didn't find the nuclear membrane that exciting or interesting. And I think, came to the conclusion that it can't be that crucial because prokaryotic organisms don't have a nuclear membrane. They survive just fine without it. In the C tier, you've got your boring organelles. Endosomes, your vacuoles, your vesicles. You've got some smooth endoplasmic reticulum. That's more boring than the rough endoplasmic reticulum, so had to go in the C tier. Nucleolus, I also put in the C tier just because it's not the nucleus, is it? In the B tier, we've got your slightly more fanciful organelles, right? You've got your rough endoplasmic reticulum, you've got your food vacuoles and your contractile vacuoles. They had to go in the B tier because basic vacuole was in the C tier. If you've got a function that makes you better than basic, you've got to go in the tier above. Ribosomes, important, in the B tier. The A tier, that's where we had organelles that were, we're getting on just the funky side, right? The funky side of organelles. Can you describe an organelle as funky? Well, I'd say the Golgi is a funky organelle. It's got a cool structure, right? It's got a funky structure compared to all the other organelles. The Golgi, recognizable immediately. Just put a picture up of the Golgi apparatus and I go, oh, that's the Golgi apparatus. See, look, you love the Golgi apparatus. There you go. Deserves its place in the A tier. So I'd say, why do you love the Golgi apparatus so much? Tell me, enlighten me. The Golgi function-wise, I don't think it's probably the most exciting of organelles, but it has the coolest shape. So it gets a lot of bonus points for that. That's why it's a funky organelle. It's got that cool shape. You've got peroxisomes up there. They're more fun than vesicles because they destroy things. They've got that peroxide in them. And it looks awesome. See, exactly. The Golgi apparatus gets these bonus points because it looks cool. I'm glad I'm not the only person that thinks that. And then the cytoskeleton, an A tier organelle, because your cell would fall apart without it. But then, getting down, we've made it to the S tier organelles. I'm going to zoom back in because these ones, they deserve their moment in the sun. Because I'd say being an S tier organelle, no, because data shape's manipulatable. I can just play around with my data. If I was an unethical scientist, which if you saw my PhD and looked at the size of those error bars, you can clearly say that my data is not made up. But you can just, I don't like how this data is looking. I'm going to change it to a logarithmic scale. And now it looks nicer. Whereas the Golgi is what it is. It has an identity and it's proud. It's proud to have that funky shape. But it isn't an S tier organelle because it doesn't have that really exciting function. It's just it's a sorting organelle. It's not a cell membrane. It's not your phospholipid bilayer. It's not your semi-permeable membrane. So it's not the cytoplasm where everything is in the cytoplasm. It is the world of these cells. It's not the nucleus, the brain of the organelle. It's not a lysosome. Yes, lysosomes are in the S tier because I'm an immunologist. I look at host pathogen interactions, specifically Salmonella, and I've done a little bit of Staphylococcus and E. coli. And these bacteria, if you go back in the stream, I don't know how that works, but they utilize the lysosome for their own benefit. The cell is like, haha, here's an organelle that's going to destroy you, and then the Staphylococcus is like, I love a low pH. Yay, give me this lysosome. And then the lysosome comes in there. Yes, it will kill a fair few of those Staphylococcus. But those Staphylococcus that die, they weren't strong enough. Those Staphylococcus that live, and use the lysosome for their own benefit. Amazing. And the lysosome, S-tier organelle. The mitochondria, of course, it's an S-tier organelle. It's the powerhouse of the cell. That's what everyone knows when you think about organelles. What do you know about the cell? It's got a mitochondria in it. What does the mitochondria do? It's the powerhouse of the cell. Brilliant. You know, it evolved from a bacteria. That's what I was talking about earlier. That's great. And then mitochondria, the girl boss of the cell. Girl boss is probably a perfect description because, I mean, it's got that the mitochondrial DNA, right, is only passed down from mothers, right? It's maternal how the mitochondrial DNA is passed down. So it is the girl boss of the cell. You're absolutely right. The powerhouse, the workhorse, right? That often, I wouldn't say it's unappreciated because it's going up here in the S tier, but without the mitochondria, just without girl bosses of the world, we know we need more of those. So mitochondria, S tier organelle, and then last but certainly not least, you've got chromosomes, an S tier thing in the cell. You wouldn't have a cell. You wouldn't exist. Nothing. There would be no life if there was not for DNA. Yes, okay, you can have RNA-based life-ish. You know, ribosomes are made out of RNA, but they're not life. You can have RNA viruses, but are viruses alive? There's significant debate on that. I err on the side of no. Viruses are not living organisms. They're just things. They are a thing, like a rock. A rock is not a living organism. It's just a thing. A virus is a thing, a microscopic thing that is very bad for you. But I suppose if you ate rocks, that would also be bad for you. Internalizing viruses, bad for you. Whereas chromosomes and then their storage form as chromatin, life. You know, we're always on the hunt. We're always on the hunt for DNA, right? For extraterrestrial life. Would that life be? What sort of DNA would they have? The fact that new genetic treatments, you know, involve manipulating this DNA to help solve mutations. You know, DNA is who we are. Can't say any more than that. We are who we are because of our DNA. And so DNA has to go in the S tier. I wouldn't be sat here putting DNA in the S tier if it wasn't for my DNA. Wow, imagine that. What a big picture thought. So there we have it. The ranking of all the organelles of the cell, plus some bonus things that weren't organelles. So that brings this stream to an end, really. This was the first stream on Science After Hours. Thank you for those who attended. Much appreciated. Do I need to come over and make sure I'm okay? I'm getting all lovely. Don't worry, Spectre is here to look after me, to hurt me in some fashion and bring me right down to earth. She's probably getting hungry. I haven't given her a snack in a while. I'll be getting a scratch from a cat to bring me right back to my miserable ways. I'm glad you found it amazing. I don't know what the schedule for these will be at the moment because I haven't started my postdoc yet. Lots of free time. Lots and lots of free time. So who knows? Maybe I'll find something else to make a ranked video of, or some other thing to stream that is sciencey in some way. So keep an eye on the YouTube channel for that. Because I have all this free time, I've actually been working on a new YouTube video. It's about rhabdoviruses. So keep an eye out for that as well. It's been a long time coming. It's based on a film that I watched way back in May. It's the end of July now, so I had to watch a recap video of that film. I don't want to spoil the video for you, but it should hopefully be out some point next week. I think I need to make up some. I'm currently in the editing. The sound is the audio is done. The research is done. Just the editing to do, make it all good. So then it can go out there in the wild world, and then you can be the president of my fan club. That's much appreciated. There might be some competition for that role, but competition, much like evolution, breeds the best. So you can fight amongst all of my other fans to be the best fan. And then that means I have better fans. And that again feeds my ego, which makes me a better scientist, I think. Good scientists often have big egos. But I don't want to have an ego big enough that I become a scientist that nobody likes. You know, we've all, throughout academic pursuits, I'm sure we've all had lecturers or mentors in some fashion that think far too highly of themselves. Don't want to be one of those people. So we'll keep my ego in check. But thank you all for coming. Well, this was the first stream, as I say, on Science After Hours. I'm glad you enjoyed it. Hope you learned something about myself and about organelles today. And I will see you soon. Thank you very much. And now I need to figure out how to turn this off. What an exciting end to the stream. Bye bye.